So about my background, I did my schooling in a small village in Kerala. It's called Konni. It's a beautiful location. It's famous for touristic reasons. And after my schooling, I did my college and masters in University of Kerala, about 40 kilometers from here, at the Karivattam campus, where I did my masters in zoology. Okay. And then after that, I went to National Center for Biological Sciences in Bangalore, NCBS, for my PhD. Okay. My PhD was on immune cells. We were interested in looking at how the immune system functions and how are the excess immune cells removed after an immunological reaction, right? So how the excess cells which are made during an immunological reaction has to be removed from the system, and we were basically looking at how that happens. So apoptosis, basically in the peripheral system. My PhD was mainly in using mammalian model system, mouse model system, and cell lines and stuff like that. And at that point of time, I got interested in looking at developmental processes, and I got interested in looking at developmental apoptosis. I thought, and then I came across, because NCBS has very strong fly groups at that point of time, I got exposed to the fly system because of talking to people and then also interacting and working with them. So I got really interested in the fly field, and then I switched from the mammalian system to a fly field because that was a better developmental model system, and I could address some of the questions which I were interested in at that point of time. So after my PhD, as I said, I went for my postdoc. This was with Dr. Stephen Cohen at European Molecular Biology Laboratory, EMBL. He's a highly accomplished scientist who has done a lot of initial work on Drosophila limb development and cell signaling which goes on during early development of organisms. So I was quite fortunate to get an opportunity to work with him. During that time, I was supported by the German Research Foundation, and also there is another organization called the Humboldt Foundation. So I was funded by these two organizations during my stay in EMBL. My supervisor, my mentor, actually moved from EMBL, which is in Germany, he wanted to have an Asian experience. So he moved from Germany to Singapore. So we were working at the Temasek Life Sciences Laboratory, which is a part of the National University of Singapore, in Singapore. And then within Singapore, we moved to another institute called IMCB. So I've had my postdoc in 3 different places with the same person, asking the same set of questions. Basically, what I did during my postdoc was using the fly system. We tried to understand the functions of these small molecules called microRNAs. So when I started off, we knew that there are these non-coding RNAs called microRNAs, but we did not have much of an idea of what these microRNAs do, and we knew what they're molecularly supposed to do. They are supposed to regulate gene expression, but we didn't know exactly the biological functions of various microRNAs. So my idea was to go in a classical genetic way to create mutations of these microRNAs and then try to analyze the functions of these microRNAs in various contexts based on the phenotypes that you see. I did 2 major work in this aspect during my postdoc, which was on the same microRNA but at 2 different stages of development of Drosophila. And we found 2 different things in which this microRNA plays an important role in regulating gene expression, which is important for development as well as in the adult stage for maintenance of nutrient homeostasis and stuff like that. MicroRNA regulates very important genes important for Drosophila development, and then an important gene which is important for regulating insulin synthesis in the adult stage. So in these 2 contexts, one in development and one in the adult organism, we found very important target genes which are regulated by this microRNA. This was the first time that this kind of interaction was studied. Another major impact of my study was we showed that the microRNA can regulate a target gene expression, and this target in turn would regulate the microRNA expression. So this acts like in a feedback kind of mechanism in which the initial signal would, based on the second signal, can turn on or turn off the initial signal again. This was again the first time such an interaction between a microRNA and a target gene was identified at that point. What sparked my interest in science? I remember that when I was in 3rd or 4th standard itself, my teacher in the class asked me what I want to become in future. I remember my answer very clearly. I wanted to become a professor at that point of time, and I wanted to teach students science. This was very clear. And because I had support and a lot of feedback from my family, which paved a very important stage in my career. So my father used to buy a lot of books, and then a lot of them were about science. One of the early books which I got in science was actually not a simple book, it was actually an advanced book which was written by Yakov Perelmo, Principles of Physics and the Interest in Physics. So I had very early on, from in 3rd standard or 4th standard, I read his Yakov Perelmo's book. And then there were a lot of interesting books which came out in terms of science and dinosaurs and stuff like that, which again became very fascinating for me at that point. From the very beginning, my orientation was very clear. I wanted to do science. I really got interested in literature. I read a lot of books, both in my mother tongue as well as in English. And then that sparked a lot of interest in literature. So in between, I thought I should go into a career which is teaching literature, because we also had some very good teachers in literature in my school and college at the time. I probably would have never wanted to become a writer, but I wanted to read other people's work and then try to criticize that or present in a different way to people. Then another thing which I got interested was politics, do good to the society, which was basically not a great idea. I got interested in the kind of politics which will benefit humanity, and then very soon I realized that it may not be a viable option. These are the two deviations which I had, but they were only for a short time. I didn't pursue it actually. So basically my lab here works with the Drosophila model organism, and we are trying to understand how changes in the environment, basically, right? So you have a lot of fluctuations which can happen in the environment on a daily basis or even an hourly basis, right? And seasonal changes are also there. So we are trying to understand how changes in the environment is perceived by an organism, and then what kind of buffering mechanisms would be present inside the organism which can actually stabilize the internal environment. So if you have a system which is changing with the external environment, that will become chaotic, right? So many things won't function normally. And we know that biological systems has this ability to buffer these changes and remain at a stable state as much as possible. Now, it may not be very stable, but it will be stable in the sense that it will be a functional stability. So that is the rough outline of what we do. So we try to understand how organisms perceive changes in the environment and how to maintain stability, internal stability. We basically look at one aspect, because we can't work on everything. So we look at one aspect, which is the change in availability of nutrients, right? Because all living organisms require nutrients for their survival, growth, and functions. And this is something which in nature would fluctuate quite a bit. The availability of food is like something which can change quite a bit. It doesn't affect humans much, or which in humans it can be controlled in various ways, because we are a developed society, developed organism. We control what we eat, and that's possible. But for an organism which is living in the wild, in nature, these things are not possible. So they should have mechanisms by which the body adapts to availability of food, and also to find food and search for it, even compete for food, and while performing other bodily functions as well, right? So how does food availability or nutrient availability is regulated in the organism in a way that even small changes doesn't affect it. And when you have plenty of food available, then how do you store it and use it for later? So these are the things which we are trying to understand using the Drosophila model organism. So that is the basic interest of the lab. We use a lot of genetics, we use cell biology techniques, we do a lot of imaging using different types of microscopes which we have in the institute, and we do a bit of biochemistry and molecular tools as well to address these questions. Okay, so that's basically what my lab does. So I have 5 PhD students in the lab, and right now I have 3 master students who are working with me this year. Normally I have 3 to 5 students working every year from the masters this thing. So far it's a great experience, because you teach, you train, and you kind of mentor students. That becomes a really nice experience, and that's quite nice and unique, which I'm enjoying after I've joined here. So basically we do research for 2 things. One is to understand things better, right? How does anything work? How does anything function? Is one of the basic questions which we ask, right? And a lot of this is basically curiosity driven, and a lot of questions needs to be addressed there, because we still don't understand. Unlike probably physics, chemistry areas. In chemistry, I think a lot of basic work is already done and understood. In physics, probably there's a lot more questions which in the cosmos and universal questions are still remaining, but a lot of things are understood to some level. Whereas in biology, even now, I think even after many years of research, there are still things which remains to be understood. These are sometimes basic fundamental questions which are not understood. The text of genetics which I studied during my PhD had 10 additional versions from that time till now. And this is not because they are printing new versions, they are printing new versions with additional information. So a classical field like genetics has changed so much in past 15 years, right? So that biology is a dynamic field which is rapidly changing. That's one reason which is exciting to work in biology. Okay, that's my main drive. Let me accept that, and that's the reason why I am in biology and why I do biology and why I do science is because I'm trying to understand more and more about living things. We have to accept that whatever we do should also have an applied aspect as well, which is very important because it's a part of being a good human being, part of being a good social animal. You have to do something which is beneficial to things around you, which you can do directly, or sometimes you can do by trying to understand how systems work. You're trying to understand how internal systems are maintained at different conditions. A loss of this balance in any context, in humans or in lower organisms, can be very bad. In non-human animals, I think normally if your nature and if your balance is affected, I think normally you may perish just quickly because you can't survive. A lot of ecological pressures are there which will kind of get rid of you. But in the case of humans, for example, these balances would lead to things like disease conditions or disorders. For example, you have excess growth, you have cancer-like conditions, you have immunological consequences and stuff like that. All this require a balance in the system. You need to have the right amount of neurons in your brain to be able to function normally. And the cell number in a system, in a particular organ, should be maintained in a certain way. And if you have too many of that or if you have too less of that, this can affect the normal functioning of a biological system. The question which we ask about how the internal balance or internal homeostasis is maintained can be used to address any of these problems, basically. And if you talk about model systems, like, even between mouse and humans, there's a huge difference. Even between chimpanzee and gorilla and humans, there's a big difference. So the things are very different between these systems. So I think even if you have a large gap between fly and humans, I think the basic understanding of how these things function can be applied across organisms. I think that's what I feel. That's an interesting question. A typical day at work is, I should say, it's quite busy. You have to manage quite a few things. You have to manage your research and you have to talk to your students, the research students, which I do. Like, I try to meet at least one student per day kind of thing. And then we have lab meetings in which one of the students would present their work and stuff like that. Apart from that, for every lecture, different lecturers have different styles of preparing for a lecture. So I know that some people prepare and then it's fine. They remember everything and they're ready to go at any point of time. But for me, every lecture means I prepare 6 to 8 hours for every lecture. That is probably not an efficient thing to do. But as a result of this, I update a lot myself, and then I feel more happy and confident about what I'm giving the class during every lecture. So I try to give them something more than what I gave last year. So a lot of time is gone into teaching, preparation of teaching, and then what you taught and teaching hours, then meeting students, and those things are also there. Then I have quite a bit of administrative work. Most of them involves interacting with students again. So I'm a part of the placement cell. I'm a part of the health center committee. So I'm also a part of various administrative committees and stuff like that, which is also very important because our institute is still in a developing phase. We are in the early phase. Contributions from people who are willing to contribute is really important for, in the end, making a very good institute. We need to be seen as a very efficient, very good institute where people would want to come and visit and do good science. And I try to contribute in whatever way I can, even though it's very small, whatever way in which I can to take care of it. So a typical day is busy. It starts sometimes it starts from home itself at 7 in the morning. It goes on till 10, 10:30, 11 in the night. And I spend at least from 9 till 7 in the evening in the lab in my office. But it's fun, it's challenging, it's tough, but I'm enjoying it. And that is the most important thing about what you do. The science is all about trying out new things. If you were doing which has been done by other people, then there is no point in doing science, right? You're always exploring new things. So you have to establish new things, new protocols, new experiments, new research programs, new questions has to be asked at every point of time. Maybe even on a daily basis, you would be like thinking about and refreshing your own thoughts and stuff. So that is very challenging. That challenge is interesting, that keeps you going. But then the problem is sometimes experiments don't work. You had a plan, you had a hypothesis, you had this idea, you tested it again, doesn't work. What do you do? Someone told me that 95% of things don't work in science and in biology especially. And then you should think that we have this very complicated systems where you have to keep model organism alive and function normally. You have to put all your efforts into them functioning normally. Then you perform a very difficult experiment. You do like all kinds of molecular, biochemical readouts or behavioral readouts, and in the end it doesn't show anything. Right? That can be very frustrating. But it also tells you that something went wrong at some point of time, and then you can try to correct that. Maybe your hypothesis was wrong, that's also possible. Right? Or maybe you haven't asked the right question. So all those things can be dealt with, but if something went wrong in terms of things which you couldn't control, that becomes frustrating. So that's where getting the right things at the right time. For us, it's a challenge to get the flies from labs abroad in time in India because of some customs regulations. So this is some issue which even we are kind of negotiating with the higher administrative bodies. Even the Prime Minister's scientific advisor is helping us with that. So we have talked to him, and we are trying to figure out a system in which we can get the scientific material, especially flies, from labs abroad without too much trouble in India. So that is one huge limitation which we have, and that's the biggest source of frustration for us right now. Not getting things at the right time. Though I may look like a person who has a lot of patience, I am actually not. But I don't show that. I become very impatient, but I try not to show it because there's no point in spreading out your level of impatience to someone else. Right? So you become impatient in a certain situation, but you try to get back to normalcy as quick as possible, because being impatient all the time won't help you, because you can't think straight when you're impatient. Your thoughts won't be normal. Your interactions with other people also won't be normal. You'll get frustrated and angry with people like unnecessarily. So remaining patient is the best thing which you can do. But in a given situation, you won't reach that mode very easily. You may have some fluctuations, but eventually I think try to reach there as quick as possible is the best thing. I have a lot of hobbies. I am interested in various things, but I have a disclaimer to make in the beginning. If you ask me like what I do these days, it's apart from work and apart from lab and teaching, I pretty much do nothing. Okay? Because of the limitation of time and because of some challenges which we are facing right now, which requires all my time to be focused on work. But this phase won't continue forever, I know that. And I'll again find time to do other things as well. But I have many hobbies. My most important hobby is reading. I read a lot, fiction and non-fiction, sometimes politics, history, a bit of history, a bit of human behavior. Then I have a large collection of books. I like to watch movies. So this is something which I do even on a daily basis almost. I try to watch like 20 minutes of some movie every day. So that helps me somehow. That helps me to cool down. Even on a busy day, I would try to watch. I used to travel a lot. In fact, I've traveled to some 22 countries. I like to try out foods from different regions. I'm a foodie, but I don't eat much. I don't eat a lot, but I like different types of food. Sometimes I do cooking. So I think these things keep me engaged. So I spend time with my daughters. I have 2 daughters. I spend time with them, and then they also keep me engaged, and I don't need to do a lot beyond this to keep me happy, because spending time with daughters itself is fantastic. We should be able to tell what we are doing. I think we should tell others what we are doing. I think that's very important. Science communication is really important. What we do in the lab and what we do as a scientist. Because from an outside person, they will look at you and then they won't know what you're actually doing, right? So they see this huge structure, lots of money being spent here, but they don't know what comes out of here. Training. We are teaching students. We are sending out about 200 students every year who are well trained in science. That's the biggest thing which we do. And then these kind of interviews and these kind of platforms would help people someone else who may not have any clue would find this interesting, and they may actually get attracted towards science because of these kind of interactions, right? They will know that a scientist can be a normal human being, right? Because a lot of people think that scientists are like someone special. They're in this bubble, and we have no relations to them and stuff like that. It's actually not true. We are all normal human beings, right? So this kind of interaction will help them to tell them that we are kind of normal people.